Welcome back to the deep dive. Today, our mission is, well, to journey right into the core of acute inflammation. We're exploring that final, crucial phase, how the immune system's specialized cells actually find an invader, capture it, chemically dismantle it, and then meticulously clean up the battlefield.
Yeah, the cleanup operation.
Exactly. We are diving deep into the body's ultimate cleanup crew, this really complex and kind of double-edged process of phagocytosis and clearance.
This is really where everything we've discussed about inflammation, converges. If you think back, we talked about the vascular changes, the dilation, the permeability,
Right.
and then the leukocytes, the white blood cells getting out of the vessels. Emigration.
Okay.
Phagocytosis is, well, it's the main way these specialized cells, mostly neutrophils and macrophages, actually eliminate the offending agent.
And that agent could be a microbe or maybe just dead tissue?
Exactly, either one. Dangerous microbes or, necessary removal of necrotic host tissue.
So we've kind of established these three huge parts of acute inflammation. The vessels changing, the cells getting out,
Immigration.
and then this final elimination step. And if this part fails, the whole defense just stalls out, right? That could lead to chronic problems.
Absolutely. Without effective clearance, that acute process, it can just spiral. You get persistent inflammation, tissue damage, maybe even fibrosis down the line. So this process, it's highly regulated, incredibly intricate, and, well, essential for survival.
Okay, let's unpack this then. Starting with the journey. How do the cells even get to the site of injury? Because moving these cleanup vehicles from the bloodstream highway to a specific spot, that sounds like some serious molecular engineering.
Right. So the first change is actually physical. It's all driven by those vascular changes we mentioned.
Okay.
When the vessels dilate, the blood flow slows way down. We call that stasis.
Like a traffic jam.
Exactly like a traffic jam. And this slowing, it forces the red blood cells, which normally flow in the center, to kind of bunch up. And that pushes the lighter white blood cells, the neutrophils, the monocytes, pushes them out of that central stream towards the inner surface of the vessel wall.
Ah, okay. So they get nudged to the edges.
That redistribution, that's called margination.
So they're effectively bumped from the fast lane to the shoulder, but once they're near the wall, they can't just, slam on the brakes, can they? They need to start interacting gently.
Precisely. They need to slow down first.
Yeah.
This leads us to the crucial rolling phase.
Rolling. Oh.
Rolling is all about weak, transient, sort of low-affinity sticking. It's mediated by a family of adhesion molecules called selectins.
Selectins?
Yeah, there's L-selectin on the leukocyte itself, and then E-selectin and P-selectin on the vessel wall, the endothelium.
And how do those E and P selectins get there?
Good question. Local chemical signals, mainly cytokines like TNF and IL-1, they act on those endothelial cells lining the vessel, and they cause them to quickly put these E and P selectins onto their surface.
So the cytokines are like the signal flare saying injury here.
Exactly. And these selectins function like molecular hooks, maybe? Or maybe Velcro is a better analogy.
Okay, Velcro. I like that.
They're carbohydrate-binding molecules, lectins, technically, and they grab onto specific sugars on the leukocyte surface. But the bonds are weak.
So they catch and release.
Right. The blood flow keeps pushing them, so they attach, detach, literally rolling along the surface.
Like a wheel slowing down.
Yeah, it's the essential first braking step. If they couldn't roll, the blood flow would just sweep them right past the injury site. They'd miss it completely.
Okay, so that slow rolling has to become a dead stop. How does the body make that shift from just, weak sticking to firm adhesion where the cell is really locked down?
This is, I think, one of the most elegant bits. The transition is triggered by different chemical signals called chemokines.
Chemokines. Different from cytokines.
They're a special type of cytokine, really focused on attracting cells. A key one here is IL-8, especially for neutrophils. These chemokines, they're produced right at the injury site, and they get displayed, kind of like little flags, on the surface of the endothelial cells.
So the rolling cell bumps into these chemokine flags.
Essentially, yeah. As the rolling leukocyte passes over them, the chemokine binds to a specific receptor on the leukocyte surface. It's a G-protein coupled receptor.
And that binding is the stop signal.
It's the activation signal, really. This binding triggers a super rapid, profound change inside the leukocyte. We call it inside-out signaling.
Inside out.
Yeah, the signal from the outside receptor tells the inside of the cell to change something on the outside surface. In this case, it targets the cell's main adhesion molecules. the integrins.
Integrins, okay, like LFA1.
LFA1, MAC1, those are the key ones on leukocytes. Normally, they're in a sort of bend-over, inactive, low-affinity state.
Right.
The chemokine signal causes them to instantly switch conformation. They straighten up, extend out into a high-affinity state.
Wow, that sounds dramatic, like flipping a switch.
It really is, almost instantaneous. And once that integrin extends, it grabs hold tightly to its partner molecule, its ligand, on the endothelial cell surface.
Like ICAM1.
Exactly, ICAM1 or VCAM1. These bonds are strong enough to resist the sheer force of the blood flow. That stops the cell completely. Dead stop. Now it's ready to get out.
So, selectins for rolling, chemokines trigger the switch, integrins lock it down.
Okay.
Now it's stuck to the inside wall. How does it physically get out of the vessel? That's transmigration or diastasis.
Yeah.
How do these relatively big cells squeeze through the vessel wall without, ripping it open and causing bleeding everywhere?
They're remarkably flexible. They mostly squeeze between the endothelial cells right at the junctions where they meet.
Not through the cells.
Generally not, mostly between them, especially in the postcapillary venules where the vessel wall is thinnest and the junctions are a bit looser.
Okay.
And they use another adhesion molecule here, CD31, also called PCAM1. It's on both the leukocyte and concentrated right at those endothelial junctions.
Like a zipper or a handshake?
A handshake is a good way to think of it. It helps guide the leukocyte through that gap. Then they secrete a little bit of enzyme to temporarily digest a tiny path through the basement membrane underneath the endothelium.
And the gap seals up behind them?
Usually, yeah. The endothelium reseals pretty quickly. It's a very controlled exit.
Okay, they've made it out. They're in the tissue now. But the tissue isn't empty, right? It's swollen, damaged, full of other cells. How do they navigate this, 3D maze to find the actual microbes they're after?
That takes precise navigation. It's called chemotaxis. Locomotion along a chemical gradient.
Following a scent trail.
Exactly. They follow the trail right to where the concentration of the attracting chemical is highest, which is usually where the microbes are clustered or the damage is worst.
And what chemicals make up this scent trail? What are they actually smelling?
We can group them into two types. Exogenous, meaning from outside, usually the microbe itself. Bacteria, for example, make proteins that start with a specific amino acid tag. N-formyl methionine. Our cells don't use that tag, so our phagocytes recognize it as foreign.
A direct bad guy signal.
Right. Then there are endogenous factors, made by our own body in response to the injury.
Ah, the alarm signal.
Yeah. High concentrations of those chemokines we mentioned, like IL-8, also a really potent piece of the complement system called C5a, and metabolites from arachidonic acid, specifically leukotriene B4 or LTB4.
LTB4, okay. So these chemicals bind to the surface of the cell. How does that translate into physical movement? Does the cell just start crawling?
It's pretty amazing cellular mechanics. When these chemoattractants bind to their receptors again, often G protein-coupled receptors, it kicks off signaling inside the cell that dramatically reorganize the internal skeleton, the cytoskeleton.
The actin and myosin.
Exactly. At the front edge of the cell, the side facing the highest concentration of the attractant, it rapidly builds up actin filaments, polymerizes them.
Pushing the membrane forward.
Right, pushing out little extensions, pseudopods. At the same time, myosin filaments gather at the back, the trailing edge.
To pull it along.
Yeah, they contract and pull the rest of the cell forward. It's like the cell is literally pulling itself towards the signal. The source material calls it like front-wheel drive.
Front-wheel drive, I like that. That ensures they don't waste time wandering around. Now, you touched on this earlier, but let's talk about the types of cells doing this. Is it the same cleanup crew the whole time?
No, there's a very important predictable shift over time. You can see it on graphs, actually. Neutrophils absolutely dominate the first wave.
The shock troops.
Right. Usually the first 6 to 24 hours. Why? Well, they're more numerous in the blood. They respond faster to those initial signals, the selectins and integrins. And they come preloaded with granules packed with killing stuff, ready to go.
But they're not built for the long haul.
Not at all. They're ternally differentiated. They're designed to be short-lived. Once they get into the tissue, they typically undergo apoptosis, programmed cell death within hours to maybe a couple of days. They put out the immediate fire.
So who takes over then?
They get replaced by monocytes. These monocytes circulate in the blood, but when they enter the tissue, they mature into macrophages. This usually happens around 24 to 48 hours into the inflammation.
And why are macrophages better suited for the sort of sustained cleanup?
Several reasons. They live much longer. They can actually divide and proliferate within the inflamed tissue, unlike neutrophils.
Ah, they can call in reinforcements locally.
Sort of, yeah. And critically, they're not just about eating and killing. They're also major players in tissue repair and remodeling. They're the ones that engulf the leftover debris, the dead host cells, and importantly, the dead and dying neutrophils themselves.
So the type of cell you see tells you about the timing.
It's a key indicator. Lots of neutrophils, probably early, acute inflammation, mostly macrophages, suggests it's been going on longer or it's starting to resolve.
Okay, so our phagocyte, let's say it's an early stage neutrophil. It's navigated the traffic, squeezed out of the vessel, followed the LTB4 trail. It's arrived. Does it just start eating immediately, or does it need one final go signal?
It needs activation specifically for the task at hand. The recruitment signals, the chemokines, got it there. But now it needs recognition signals to prime the destruction machinery.
Okay.
Once the leukocyte actually recognizes the offending agent, maybe it bumps into a bacterial cell wall or detects a damaged host cell membrane using receptors like toll-like receptors, TLR.
VLRs, right, pattern recognition.
Exactly. That recognition triggers internal signaling pathways. Often involves a jump in cytosolic calcium, activation of enzymes like protein kinase C, and these signals are what really kick off the phagocytosis process itself. They start assembling the killing machinery.
And how does the cell know what to eat amidst all the chaos? What says, eat me versus leave me alone?
It uses several types of specialized phagocytic receptors on its surface. One really important group is the mannose receptors.
Mannose. Why mannose?
Because these receptors recognize specific sugar patterns, mannose, and fucose residues that are common on microbial surfaces.
But not usually on our own healthy cells.
Right. Our healthy cells usually cap those sugars with salicylic acid. So the mannose receptor is a good way to spot non-self directly.
Okay. What else?
There's another broad class called scavenger receptors. They were first found binding to, like, modified LDL cholesterol, but it turns out they also bind to a whole bunch of different microbial molecules and also to our own cells that are undergoing apoptosis, programmed cell death. So they help clear debris too.
So there's direct recognition, but I remember reading that's not always super efficient, that the body uses something to make the targets, like, tastier.
Exactly. To really ramp up efficiency, the body uses opsonization.
Opsonization.
Yeah.
Coating the target.
Literally coating the particle, yeah. It provides high-affinity handles that the phagocyte can grab onto really easily. Trying to eat a non-opsonized particle can be slow, kind of clumsy.
But an opsonized one.
It's almost guaranteed. Fast, efficient engulfment. It's a huge advantage.
So what are these molecular handles, these opsonins?
There are three main systems we rely on. First, IgG antibodies. If you've encountered that microbe before or been vaccinated, your body makes specific IgG antibodies that stick all over it.
And the phagocyte has receptors for IgG.
Yep. Fc receptors that grab onto the constant Fc part of the IgG molecule. That's a major link to the adaptive immune system.
Okay, what else? For maybe the first time you encounter something.
For that, the complement system is crucial, specifically a fragment called C3b. When the complement cascade gets activated, which happens quickly on many microbial surfaces, it deposits loads of C3b onto the target.
Like little flags.
Exactly. And phagocytes have complement receptors, CR1 and CR3, that specifically bind to C3b. This is critical for innate immediate defense.
So C3d is the immediate nonspecific tag, while IgG is more targeted from past exposure.
Generally, yes. C3b deposition can happen very rapidly via the alternative or lectin pathways, even before you've had time to make antibodies. It's the innate system's way of marking things for immediate removal.
Is there a third type?
There are also some plasma lectins, like mannose-binding lectin, MBL, which can bind to microbial surfaces and act as an opsonin, often by activating the complement system itself.
Okay, so the target is recognized maybe directly, maybe via opsonins like C3b or IgG. It's attached. Now the cell has to actually swallow it. Engulfment. How does that physically work? It's not just passive absorption, right?
Yeah, definitely not passive. It's a very active, dynamic process, powered heavily by the cytoskeleton. The cell membrane flows outwards, extending these projections called pseudopods.
The false feet?
Right. They wrap around the particle. It's often described as a zipper mechanism.
Zipper.
Yeah, because it seems like receptors on the phagocyte membrane sequentially bind to ligands all over the particle surface, pulling the membrane tightly around it, like closing a zipper.
And that requires
intense actin polymerization right at that site of contact. Building those pseudopods takes a lot of cytoskeletal rearrangement.
Okay, the zipper closes, the membrane pinches off, and the microbe is trapped inside.
Correct. Trapped inside a membrane-bound vesicle. That vesicle is called the phagosome.
But the phagosome itself isn't the killing chamber, is it?
No, the phagosome is just the holding cell. The real action starts when the phagosome quickly fuses with another type of vesicle inside the cell, the lysosomal granules. These are sacs packed with destructive enzymes and chemicals.
Ah, the weapons cache.
Exactly. When the phagosome and lysosome fuse, they form the phagolysosome. And that is the specialized compartment where the chemical warfare really begins.
Okay, here's where it gets really interesting.
Right.
We've caught the intruder, locked it in the phagolysosome. Now the cell unleashes its arsenal. Let's start with probably the most dramatic weapon, using oxygen itself, the respiratory burst to generate reactive oxygen species, ROS. That sounds incredibly aggressive.
Oh, it is. It's like setting off a controlled chemical explosion inside the cell. The respiratory burst name comes from the observation that right after phagocytosis starts, the cell suddenly starts consuming oxygen at a massively increased rate, hundreds of times faster.
Wow. And what's driving that oxygen consumption?
It all hinges on a crucial enzyme complex called NADPH oxidase. Sometimes it's called phagocyte oxidase.
Okay, NADPH oxidase. How does it work?
It's actually a fascinating piece of machinery. It's made of several protein components that are normally separate in the resting cell, but upon activation.
Triggered by the phagocytosis.
Right, they rapidly assemble together, specifically on the membrane of that newly formed phagosome.
Ah, so the weapon is built right where it's needed.
Exactly. Once assembled, NADPH oxidase takes electrons from a molecule called NADPH inside the cell cytoplasm, and it transfers those electrons onto molecular oxygen, O2, that's inside the phagolysosome lumen.
And what does that do to the oxygen?
It reduces the oxygen, creating the first key product, the superoxide anion. That's O2 with an extra electron, written as O2. It's highly reactive.
Sugar oxide, is that the main killer?
It's damaging, but it's mainly the starting point. Superoxide then quickly gets converted, either spontaneously or by an enzyme, into hydrogen peroxide, H2O2.
Hydrogen peroxide, like the stuff you buy at the drugstore.
Chemically, yes, H2O2. It's somewhat bactericidal on its own, but its main job in neutrophils is to be the fuel for an even more potent system.
More potent than hydrogen peroxide.
Much more. This is the MPO halide system. It's considered the most powerful bactericidal mechanism neutrophils have.
Okay, MPO halide, you mentioned bleach earlier. Is this where that comes in?
This is exactly it. Neutrophils have these large granules called azurophilic granules. They're packed full of an enzyme called myeloperoxidase, or MPO.
Myeloperoxidase.
When the phagosome fuses with these granules, MPO gets dumped into the phagolysosome. MPO is a catalyst. It takes the hydrogen peroxide, H2O2, that was just made, and in the presence of a halide ion, usually chloride, Cl, which is abundant in the cell, MPO uses the H2O2 to oxidize the chloride into hypochlorite, HOCl.
Hypochlorite, that is the active ingredient in bleach.
It absolutely is. And it's incredibly toxic to microbes. It just devastates them by oxidizing their proteins, their lipids, everything. It's a chemical kill shot.
That is astonishing. The cell makes bleach to kill bacteria. But if the system is so critical, what happens if it breaks? If that NADPH oxidase doesn't assemble properly?
That leads directly to a devastating immunodeficiency disease called chronic granulomatous disease, or CGD.
CGD, okay.
It's an inherited condition where one of the components of that NADPH oxidase complex is faulty. So the patient's phagocytes, their neutrophils and macrophages, they can still do everything else. They can roll, stig, get out of the vessel, even engulf the microbes perfectly fine.
But they can't kill them.
They can't mount an effective respiratory burst.
Yes.
They can't make the superoxide, can't make the H2O2, and therefore can't feed the MPO system to make bleach. The ingested bacteria and fungi just survive inside the phagocyte.
Wow, so what happens to the patient?
They suffer from recurrent severe infections, often with relatively low virulence organisms that a healthy immune system handles easily. And they tend to form these persistent inflammatory lesions called granulomas as the body tries desperately to wall off the infection it can't clear. CGD really underscores just how vital this ROS pathway is.
But nature loves redundancy, right? The body doesn't put all its eggs in the oxygen basket. There are also reactive nitrogen species, RNS, involved.
That's right. There's a parallel nitrogen-based system. This one relies on nitric oxide, or NO.
Nitric oxide, isn't that involved in blood vessel dilation too?
Yes, it's a fascinating molecule with many roles. But for killing, we're interested in NO produced by an enzyme called nitric oxide synthase, or NO, specifically the inducible form, INOS.
Inducible NOS, where is that found?
It's primarily induced in macrophages, usually not so much in neutrophils. It gets switched on by inflammatory signals, especially the cytokine interferon gamma, IFN and NL.
So this is more of a macrophage weapon.
Generally, yes. iNOS takes the amino acid arginine and converts it into NO gas.
Is NO itself very toxic to microbes?
It has some direct antimicrobial activity, but its real killing power comes from collaborating with the ROS system.
Ah, teamwork.
Exactly. NO reacts incredibly quickly with the superoxide anion O2 that's also being generated, perhaps by the macrophage's own NADPH oxidase or maybe leaking from nearby neutrophils.
And NO plus superoxide makes?
It makes a highly reactive and very nasty molecule called peroxynitrite. OMO-NO.
Peroxynitrite.
Peroxynitrite is a potent oxidizing and nitrating agent.
Yeah.
It damages lipids, DNA, proteins. It's really destructive to the engulfed microbe.
It's interesting the source material mentions studies in mice. If you knock out either the ROS system, NADPH oxidase, or the RNS system, INOS, the mice are only mildly more susceptible to infections.
Right.
But if you knock out both,
Then they succumb very rapidly. It clearly shows these systems are overlapping and somewhat redundant. Nature has built in backups.
so neutrophils rely heavily on the MPO bleach system. Macrophages use more of the no-peroxynitrite pathway, but both contribute ROS and RNS overall.
That's a good summary. The MPO system is maybe the neutrophil's primary sledgehammer, while iNOS gives the longer-lived macrophage sustained killing power. Together, they ensure potent clearance.
Okay, beyond these reactive oxygen and nitrogen radicals, the phagolysosome is also just filled with more traditional digestive enzymes, right? Lysosomal enzymes and other proteins.
Absolutely. Think of these as the molecular scissors and drills. A key group are the neutral proteases.
Protease is protein cutters.
Right. Enzymes like elastase and cathepsin G. These are powerful enzymes capable of breaking down tough structural components, not just in the microbe, but also in the surrounding damaged tissue. Things like collagen, fibrin, elastin, basement membrane proteins.
So they're essential for cleaning up the debris after the fight?
Very much so. They help liquefy the damaged area,
Right.
allowing for eventual repair. But
There's always a but with these powerful tools.
exactly. This introduces a huge problem of control. If you have an enzyme like elastase, which chews up elastin, the protein that gives tissues like your lungs their stretchiness, you absolutely need mechanisms to keep it contained.
Or it will start degrading healthy tissue.
Right. If these enzymes leak out of the phagolysosome, or if they're released deliberately by the cell, which can happen, they could cause massive collateral damage.
So how does the body control them? Are there inhibitors?
Yes, and they're crucially important. The blood serum and tissue fluids are loaded with antiproteases. The star player here is alpha-1 antitrypsin, often abbreviated as alpha-1 AT.
Alpha-1 antitrypsin.
It is the major inhibitor of neutrophil elastase. It's constantly circulating, ready to rapidly neutralize any elastase that escapes containment.
And what happens if you don't have enough alpha-1 AT?
That leads directly to serious lung disease. Inherited deficiency of alpha-1 antitrypsin means there isn't enough brake on elastase activity.
So the elastase runs wild.
Essentially, yes. Especially in the lungs, where neutrophils are often present due to minor infections or irritants. The unchecked elastase gradually destroys the lung's elastic tissue over years.
Leading to?
Emphysema, a severe, irreversible lung condition. It's a perfect, albeit tragic, example of how the balance between the weapon, elastase, and the shield, alpha-1 AT, is critical. Fail that balance, and the defense itself becomes the disease.
It really drives home that double-edged sword concept. Before we move to the consequences, were there other things mentioned in those granules besides proteases?
Oh yes, the granules are like little toxic cocktails. They also contain defensins. These are small, positively charged peptides that basically punch holes in microbial membranes. They're directly toxic.
Okay.
There's also lysozyme, which is famous for breaking down the peptidoglycan cell wall of many bacteria.
Like penicillin, sort of?
It attacks a similar structure, yeah.
Yeah.
And also lactoferrin, a protein that binds iron very tightly.
What?
Because most bacteria desperately need iron to grow. Lactoferrin essentially starves them by hiding the iron away. So it's a layered attack: radicals, bleach, enzymes, pore formers, nutrient sequestration. Really comprehensive.
Okay, so we've covered the classic eat and digest methods, but science moves on. Let's talk about a more recently understood and frankly quite dramatic mechanism: neutrophil extracellular traps, or nets.
Nets are fascinating, yeah. A relatively recent discovery in the grand scheme of immunology. What they are, physically, are these extracellular networks like sticky webs or nets.
Made of what?
Primarily, they're made of the neutrophil's own nuclear material. It's DNA chromatin extruded out of the cell.
Wait, the cell throws its DNA outside.
It does. It's a form of cell death, often called mitosis. It seems to be triggered by strong inflammatory signals. And importantly, it requires that ROS production we talked about earlier.
So the respiratory burst is involved here too.
It seems to be a key trigger. The ROS signaling leads to activation of an enzyme called PAD4, which modifies the histone proteins that package DNA. This causes the chromatin, the DNA structure, to decondense, to massively unwind and swell up.
Okay.
Then the nuclear membrane breaks down, and the cell essentially ruptures, spewing this decondensed chromatin, which is inherently sticky, out into the environment.
And it's not just plain DNA.
No, that's the clever part. This DNA mesh comes out coated with many of the neutrophils' antimicrobial granule proteins, things like elastase, MPO, defensins.
So it's a preloaded trap.
Exactly. It serves two main purposes. First, the sticky DNA physically traps microbes, preventing them from spreading, like a net. And second, by concentrating those high levels of antimicrobial enzymes right there in the mesh, it creates a localized zone of intense killing power. It's a way to fight infection, even after the neutrophil itself has died.
A final act of defense. A suicide mission, almost.
You could definitely see it that way. But this process, spilling nuclear contents out into the open, it has potentially serious downsides, doesn't it? Especially concerning autoimmunity. That is the major concern, yes. Normally, when a cell dies by apoptosis, its contents, including nuclear material, are neatly packaged up and quickly cleared by macrophages. Very clean.
But netosis sounds messy.
It is. You have large amounts of DNA and associated nuclear proteins, histones, suddenly exposed in the extracellular space. These are molecules that the immune system normally doesn't or shouldn't see much of.
Because they're usually inside the nucleus.
Right. And chronic exposure to these self-antigens, the DNA and histones from nets, is now strongly implicated as a potential trigger or fuel for certain systemic autoimmune diseases.
Like lupus.
Systemic lupus erythematosus, SLE, is the prime example. In lupus, patients make antibodies against their own DNA and nuclear proteins. It's thought that maybe defects in clearing away nets could lead to this sustained presentation of self-antigens, breaking tolerance and driving the autoimmune response.
So another example of a defense mechanism potentially backfiring.
It really highlights the theme. Potent defense always carries risk. Which brings us squarely to the broader issue of leukocyte-mediated tissue injury. The collateral damage.
Because the weapons aren't perfectly targeted.
Exactly. Hypochlorite, peroxynitrite, elastase. They're designed to destroy biological molecules. They can't always tell the difference between a bacterial membrane and a host cell membrane right next to it. Damage is almost inevitable sometimes.
So, under what kinds of situations does this necessary defense become significantly destructive to us, the host?
The source outlines three main scenarios. The first is just unavoidable collateral damage during a normal, appropriate response to a really tough infection.
Like TB.
Tuberculosis is a classic example. Mycobacterium tuberculosis is hard for phagocytes to kill. The body mounts a prolonged, intense inflammatory response trying to contain and eliminate it. But over time, the continuous release of toxic products from activated macrophages and neutrophils causes significant damage and scarring in the lung tissue. The response itself causes much of the pathology.
Okay, that makes sense. What's the second scenario?
Inappropriate responses. This is when the whole inflammatory cascade gets triggered mistakenly.
Like in allergies?
Allergies, yes. Like severe asthma, where leukocytes are recruited to the airways in response to a harmless pollen grain, and their released products cause bronchoconstriction and damage. Or in autoimmune diseases, where the immune system attacks self-tissues. The recruited leukocytes then damage joints in rheumatoid arthritis. Or the myelin sheath in multiple sclerosis. The weapons are deployed against the wrong target.
Okay. And the third one, it had a great name. Frustrated phagocytosis.
Frustrated phagocytosis. This happens when a phagocyte encounters something it really wants to eat, something that strongly activates its phagocytic receptors, but it physically can't engulf it.
Why not? Too big. Stuck down.
Usually, yeah. Maybe it's a large splinter or immune complexes deposited flat onto a basement membrane, like in some types of kidney disease, or maybe large crystals, like the urate crystals that cause gout.
So the cell tries to eat it but fails.
It tries. It binds strongly, activates its machinery, starts trying to extend pseudopods, but because it can't fully enclose the target, the phagosome membrane can't seal properly.
Oh,
right. The cell is getting strong. Eat me. Signals. So it activates the killing mechanisms, the respiratory burst, the granule release. But because the phagolysosome isn't sealed off inside the cell.
The toxic contents spill outside.
Exactly. The ROS, the NO, the lysosomal enzymes like elastase, they get discharged directly into the surrounding extracellular space, bathing nearby healthy tissues in these destructive chemicals. It's like the cell is firing its weapons wildly because it can't grapple its target properly.
That sounds incredibly damaging. Okay, so this whole clearance system is immensely powerful, but also inherently dangerous, which means turning it off quickly once the job is done must be just as important as turning it on. How does the body stop the inflammation, the active termination phase?
Right. Inflammation doesn't just passively peter out. It has to be actively shut down. There's some passive decay, of course.
Yeah.
Meaning the mediators themselves, the chemokines and cytokines, they mostly have very short half-lives. They degrade quickly once production stops. And as we said, the neutrophils, the main early responders, are programmed to die quickly by apoptosis anyway.
And the dead neutrophils get cleaned up.
Yes, the apoptotic neutrophils display eat-me signals themselves, and they get efficiently swallowed by macrophages. That removal of dead cells is itself an anti-inflammatory process.
But you said active termination. Are there specific stop signals?
Oh, yes. This is a really hot area of research, looking for ways to promote resolution therapeutically. One key mechanism involves a switch in lipid mediator production.
From arachidonic acid?
Exactly. Early in inflammation, arachidonic acid, AA, gets converted mostly into pro-inflammatory things like prostaglandins and especially those chemotactic leukotrienes like LTB4.
The come here signal.
Right. But as the inflammation progresses, particularly in macrophages, there's an enzymatic switch. They start converting AA or other fatty acids into a different class of mediators called lipoxins.
Lipoxins, and these are anti-inflammatory.
Yes, powerfully so. Lipoxins and related families like resolvins and protectins act as local stop signals.
How do they stop things?
They directly counteract the neutrophil actions. They inhibit neutrophil chemotaxis, stop them from coming. They inhibit their adhesion to the endothelium, stop them from sticking and getting out. They actively put the brakes on further recruitment.
A specific biochemical stand-down order. Are there also stop cytokines?
Absolutely. The resolution phase is characterized by the release of potent anti-inflammatory cytokines. The two big ones are transforming growth factor beta, TGF-beta, and interleukin-10, IL-10.
TGF-beta and IL-10, where do they come from?
Often from the macrophages themselves, the same cells that were recently killing things, and also from other cell types like regulatory T cells. These cytokines act broadly to suppress the production of pro-inflammatory cytokines like TNF and IL-1. They tell the whole system to calm down and shift towards repair.
So, if we connect this to the bigger picture, this smooth transition from full-on attack with ROS and NETs to active shutdown with lipoxins and IL-10. That's the key difference between getting rid of an infection cleanly and ending up with chronic inflammation and scarring.
That balance is precisely the definition of a successful, healthy inflammatory response. When those termination signals fail or are delayed, maybe the lipoxin switch doesn't happen properly or IL-10 production is impaired, the acute inflammation just smolders on. It becomes chronic, driven by persistent macrophage activation, leading eventually to tissue damage and often fibrosis, which is scarring. That failure to resolve is at the heart of many chronic diseases.
We've covered an incredible journey today, from the leukocyte just, bumping along the vessel wall,
Rolling.
to that decisive integrin stop, the complex engulfment, the sheer chemical creativity of using bleach and NO,
The MPO and IO systems.
that final dramatic act of netosis, and then, just as importantly, those crucial lipoxin signals telling everyone to stand down. It's amazing.
It really is. The redundancy, the sheer potency of this whole system. It's why we generally survive the constant onslaught of microbes. It's designed for aggression. But that aggression, as we've seen, inherently carries the risk of self-inflicted damage. It's a constant trade-off.
And understanding the molecules involved.
Absolutely. Understanding the details, how specific integrins change shape, what triggers the switch from leukotrienes to lipoxins, how nets form, that's directly informing the development of new drugs. Therapy is aimed at controlling the damaging aspects of inflammation without completely shutting down our essential defenses. It's a fine line to walk.
It definitely raises a fascinating question for you, the listener, to think about. Consider that neutrophil. As we discussed, it often has to essentially commit suicide through apoptosis or netosis for the host to survive the infection, a necessary sacrifice. So how does the body so precisely regulate the timing of that death? And maybe more clinically relevant, what happens when we intervene. When we use powerful drugs like the TNF inhibitors for treating rheumatoid arthritis or Crohn's disease that broadly block inflammatory signals, we know they work, but they also increase the risk of serious infections, like reactivating latent tuberculosis. It seems they might be interfering with this delicate life or death balance of the immune cells themselves. Exploring that regulation and the consequences of disrupting it is really crucial.